A very good afternoon to my dear students. And in this video, we are going to discuss about the Hershey and Chase experiment. Hershey and Chase, who is the scientist in 1952, he found the proof of DNA as a genetic material. So, the genetic material which is transferred from one generation to another generation. So this experiment revealed the hypothesis DNA is a genetic material. So Hershey and Chase, they evidenced that using bacteriophages, that is here they use T2 bacteriophage. Here this is the T2 bacteriophage which infects the bacteria E. coli. So what it happens first, this is the bacterial capsid and inside is a genetic material. Once the T2 bacteriophage infect the host cell, which is nothing but our E. coli. So first the capsid infect the bacterial cell, then it release their genetic material inside into the host cell. Okay, and after the host mechanisms which is governed by the bacteriophage. So the polymerases, RNA polymerases present inside the host cell which is directly transcribe the genes which is present in, which is released by the bacteriophage. So what will happen? So automatically the bacterial RNA polymerases that will transcribe the phage DNA. Okay. So what will happen? Further it will translated to the host, translated by the host ribosomes and tRNA, then it produces the enzymes. So finally what it happens? So step by step, once the DNA multiplication and after the whole phages will produced by the help of our bacterial cells. Okay, so next the produced DNA materials insert into the capsid proteins which is produced by the phage DNA. Then later on what will happen? The entire host cell will destroy by the replication action of phage DNA. Okay, so what the whole experiment concludes means the phage DNA when enter into the host cell which completely absorbs the mechanism of host mechanisms and it order to produce to replicate the phage DNAs, phage particles. So what it happens? So completely they the bacterial cells produced their offsprings like phages, T2 phages. So with this experiment, Hershey and Chase did one experiment. That is, they are using radio labeling techniques. So they are using S35 radio labeled atoms for phage capsid proteins. And in case of here P35 radio labeled phage DNA. Okay, here the green one is represents phosphorus labeled DNA, and the red one represent sulfur labeled protein. Okay, so this is the experimental design. So what will happen? As I told you already, the phage will first, it will infect the bacterial cell. Yes, both the sides. First they will infect the bacterial cells, and then the genetic material released, the genetic material will propagate itself, and finally it will produce the new progenies. Okay, these are all the whole mechanism. So in this experiment, what exactly it happens means, so here the DNA is labeled with phosphorus, and here sulfur, sorry, protein coat is labeled with sulfur. Okay, so what is the reason? So one, why the protein is only labeled with sulfur, why here DNA is labeled with phosphate? Yes, of course, here the proteins are made here cysteine and methionine are the sulfur containing amino acid. So in that sulfur is present, then only the protein will radio labeled with when the protein is radio labeled with sulfur, it look like radioactive. But in case of DNA, when it is radio labeled with sulfur, it won't like radio labeled. Okay, why why it means they don't have sulfur components. Okay, instead they are it has phosphates in their structures. So in such conditions it's look like radio label when it is radio labeled with P32, that is phosphorus. So Y means DNA has the phosphate bonds. Okay, but in case of proteins they don't have. So then only here they used sulfur, and here they use phosphorus. Okay, okay, and the second one is infection. So first we are use radio labeling, the second one is infection, and then now we are going to blending. Okay, blending means using blending just remove the capsids. Okay, the outer portion will be removed. Okay, now our genetic materials alone present. So the final one is the centrifugation. So after centrifugation, what will be the result? That is our experimental result. Okay, so here when the phage is radio labeled with sulfur, the result is after centrifugation there is no sulfur in cells. Why? Because the sulfur is only radio labeled the proteins. Okay, so protein where it is present, the protein is present on the capsid membrane. So here only, so when we are blending, all the outer membrane, that is our proteins are removed. So if you are labeled with sulfur, it will goes to the protein alone. Okay, so remaining what will be present here, genetic material alone here. But uh, here we won't label the genetic material, we just label the protein capsid. Okay, so the after centrifugation, the sulfur, there is no sulfur in cells. Okay, when coming to here, after centrifugation, phosphorus in cells. Okay, as we all know, we already radio label the phosphorus. So why here we radio label the phosphorus means the genetic material, that is our DNA has the phosphate bonds, so phosphorus groups, so we'll radio label here. So the what is the end product? or genetic material radiolabeled with phosphorus. Okay. So in this experiment, what they conclude means the complete inheritary, the complete transformation material is genes, that is DNA. So here is the further proof that genes are DNA. Okay. So Hershey and Chase found that when bacteriophage containing P32 labeled are allowed to infect the non-radioactive bacteria, all the infected cells become radioactive. And in fact, much of the radioactive was passed on to the next generation of bacteriophages. However, when the bacteria were infected with bacteriophages labeled with S35, that is our sulfur 35, and then the virus codes are removed by blending methods, practically to no radioactivity could be detected in the infected cells. So from these experiments, it was clear that the DNA component of the bacteriophages is injected into the bacterial cell while the protein component remains outside. However, it is injected into the component DNA that is able to direct formation of new viral particles complete with protein codes. So here is the proof that genes are DNA. Okay. So here is the complete experiment and here is the process of aid fiction. Okay. Thank you for your attention.